Hello students, welcome to first lecture of week 5. So in this lecture we would like to see that how much sequencing you should do for any project. Okay, so this is a very important question. When we aim for any genomic project, we should first know that how much data we are going to need either for a genome or transcriptome, so that you can achieve the purpose that you want to achieve. Okay, so purpose of course is to do the genome assembly, right? But it also depends upon what is your exact aim. For example, you want to generate a draft assembly of a genome, okay, which could be like small in size or large, very large in size. Okay, so for a very large genome, draft assembly can be a good idea. Okay, so that you can at least tell something about it, and it may take a lot of effort to produce a high quality genome and then complete the genome from start to end, right? Telomere to telomere. There a rough draft genome is okay, because many times this has been done, like in the case of human genome, which started sometime in 1990s and 95 and finished in so the first draft came in 2001, and then basically the complete genome came in 2022. So it took almost 20 years. So people were working with draft sequences, and it was a complicated, challenging project. Okay, but if you are dealing with a bacteria which is a like small genome, 5 MB, million base pairs in size, okay, there also you can produce a very high quality genome. But then also, if you want to like completely finish the genome, like you want to make a complete circle out of it, which is the circular DNA, then you will need a lot of efforts, like primer walking and like connecting the missing parts and like that. Okay, so that is another issue. So, and then also we have different type of reads to use. It could be long read sequencing by PacBio or Nanopore. It could be short read sequencing by Illumina or Ion Torrent, right? So there also you need to have an idea of how much you should sequence so that you can cover the genome appropriately. Okay, and there are several matrices which tells you that how good was your coverage sequencing. Okay, how many genes you could cover in that. Okay, so there are N50, BOOSCO, etc. matrices, right? And so basically, and this is, we are talking about high quality draft genomes or draft genomes, but you can also aim for like some kind of chromosomal level assembly if you know about like how many chromosomes are there. And if you want to go for techniques like Hi-C, then you need to have at least a minimal level of quality so that you can proceed with that kind of basically achievement. For example, you want to do the Hi-C. Okay, so that we will cover later. So now I will tell you that in this lecture how to estimate the amount of sequencing and what you should do to achieve a good quality genome. Okay, so basically sequencing coverage is one term that you will hear, that how much coverage I should achieve, okay, or I should aim for, right? So it also depends upon the sequencing read that you are talking. Okay, so and also like basically, for example, one genome is say 5 million base pair. A bird genome could be 1 billion base pair, 1 GB. Okay, eukaryotes could be like 2 to 3 billion base pairs, right? So that means you should achieve like you should only, if there is a technique which can like read all the 4 million or 5 million base pairs like in one go, then you read only one read, then you need only one read. Okay, but it is not possible, and reads are in short pieces. Okay, even if it is long, but it is like in terms of genomic reference, it is short. Okay, it cannot cover the entire genome. Okay, so if you are generating multiple reads from a genome, okay, so basically this is, for example, one region that you want to cover. Okay, so you have generated say read like this, okay, right, short and long, for example. So basically this way still like there could be some gaps will always remain, right, and some things you can cover up, right? And then basically sequencing quality also matters, and there are errors also in sequencing. So it is not that the base which is covered here is the correct base. So at least you should have some kind of coverage of each base pair, so that if I compare this and this 3 reads, or basically generally 10 reads or more, okay, per region, then you can say that you can call each and every base pair very reliably, that okay, in all the 10 positions in different reads that you have got from that region, you have got A, so that means that is the correct base, okay. Or it could be like 9 times you got A and 1 time you got T or G, so that means you should go for the correct one which has a higher call, which is more likely to be correct, okay. So you need multiple observations per base, and reads are not, when you basically not distributed like throughout the genome. For example, you take the DNA out from a cell, then you do there are several pipetting and library preparation, and these steps are involved, okay, and ultimately you make the library and load the library, right. So it is no guarantee that all the genomic DNA is covered in that library. It could be that some regions is missed, or basically when the DNA was randomly fragmented, some like this was fragmented, so basically this small piece is missing, okay, there is a small gap there, right, because it broke while you are sharing the DNA. So basically so that is why you should also need multiple reads, or multiple times you should cover the DNA when you do the sequencing of the same region, so that you can also cover those kind of fragments. And but still, like, there will be something that you miss due to assembly method, or due to lack of that read in that region, okay. Then basically so combining all these, you need like a good quality coverage, okay. And coverage is, as I said, it's a metric, or basically the way in which you should tell that how many times you have covered a genome, how many times you have read that genome. So if this is a region, so basically you should one, okay, right. So 1 2 3 4 5 times I have read it, okay, and this was the original strand, right. So how many times you can you have sequenced the same or basically you can say most of the DNA you have sequenced that number of time, okay. So that is called coverage. And for a new genome it may be a different number, because you do not know the genome sequence at all. But for a resequencing project such as human genome is available, high quality chimpanzee, like that, okay, or maize or wheat, right. So fairly good quality genomes are available, so you can do resequencing in this case, right, and that will require much lesser amount of sequencing and coverage, okay. So there are two things, like depth of coverage and breadth of coverage. Breadth of coverage means that if this is the genome, then you are kind of reading the entire genome and covering the reads from the entire genome. There could be some regions which could be telomeric, okay, or centromere, so, or basically highly repetitive regions. So at those places your sequencing may lag behind, right, but still you have tried to aim for the complete genome coverage, okay, from all the chromosomes and like that. And second is depth, that how many times you have read that genome, okay. So like in this case 1 2 3 4 5x coverage is there. X is the term that is used to tell that how much coverage you have done, 1x 2x 3x 4x 5x, okay. So for example, if the genome is like 100 million base pair, and you have sequenced 5 million of reads of 100 base pair size, and basically your coverage is 5x, okay, because you have read that you have generated 5 million reads of 100, so that means 500 MB, 5 million base pairs into 100, 500 MB, so divided by 100 is 5x. So basically roughly you can estimate like this, that how much is the coverage, okay. And breadth is like you have covered, say for example 95% of the genome, that means most of the regions in the genome are covered, but some 5% are still missing, and there are matrices which can tell you that what is your breadth of coverage, right. So breadth of coverage and depth of coverage are two terms, okay. Generally we are breadth of coverage, we generally you achieve it at the end, right. This number comes at the end. So basically after you have maybe done the first round of sequencing and you have done the assembly and everything, then you can tell that okay, I was able to cover the entire genome or not. But depth of coverage you have to decide before going for sequencing. Okay, so that is the first thing that you should think of. Okay, and you should think of how many X coverage you need from that genome. Okay. So basically this coverage is calculated based on genome size. So if the genome is small, say 5 million base pair, right, then for a typical bacteria, I think 100 to 200 to X by Illumina short reads is good enough, or 30 to 50 X by long reads is good enough. Okay, so this is about the bacteria, right? And then still, like if this was the bacteria circular, okay, then you will not be able to cover the complete circle, but you can say that okay, if okay, how many pieces you are getting that circle, right? Okay, so less than 100 pieces, like a kind of good number that you have. Okay, but sometimes you might have seen some draft genome which has like thousands of pieces, right? So this is one thing that you should look for. Okay, and it also depends upon the read size. Okay, smaller read or longer read, how much coverage you want to go for. So that is about the genome, but also about the transcriptome, you are more concerned about basically how many reads you want, okay, from that transcriptome. So that, because in the transcriptome the idea is to there can be two objectives. One is like you want to detect the number of transcripts, right, how many genes are expressing and what is their level of expression. That means you should have at least one transcript from each of the transcripts, okay, minimum, right, but like one you should have at least certainly more than one, and how many times that transcript is coming. So that will tell you that how much the gene was expressing. Okay, so in the transcriptome basically it is more about the depth, right, that okay, so basically transcriptome is also small. What is transcriptome? So basically from a genome there are introns and exons and intergenic region. Okay, and exonic region is typically 2%, for example, in human genome. Okay, so basically in the case of genome you are covering the entire 100%, but in case of transcriptome you are covering only 2%. Okay, so that means your amount of sequence data that you need for just covering that 2% sufficiently will be much less. Okay, so basically you can say that okay, for any transcriptome about 15 to 20 GB can give me a good estimate of the number of transcripts which are present and also their expression level, right? So okay, so basically transcriptomics, you should know that what is your transcriptome size. Based on the genome you can calculate the 2% or 3%, right, and then you can see that okay, how much you should go for the sequencing. Okay, and in the previous also, and like we have looked at this formula which is like how to calculate the coverage. So coverage is like length of read, number of reads divided by the genome size. Okay, so length of read in the previous case was say 100 base pair. Okay, and number of reads are say 200. Okay, and the genome size is say so basically it become 20,000. So basically genome size is say 40 kb, right? So basically your coverage is just half, right? So but you should go for as many as much as possible. Okay, so like 100 to 200x for a typical genome is kind of a good idea to go for, and we will also see some case studies. So basically I have used some rough numbers here, okay, right, but just to tell you that how it is calculated, but basically at this point we have taken only say number of reads only 200, but we will be talking about millions here, right? Then of course this coverage will go up. Okay, and this genome size is the haploid genome length. This is to be seen. Okay, so this, if I saying like 40 kb genome, okay, typically a plasmid or something, right? So this is in the case of plasmid, this haploid thing is not relevant, but basically for any human, for example, diploid genome, okay, or any plant is tetraploid, right, or polyploid. Okay, so basically in that case you should only look at the haploid genome length and not the not consider the polyploidy. Okay, then we have seen the short and long reads concept earlier. So short reads are can be produced at much higher depth, and they have their own purpose, they are more accurate, and long reads suffer from like some errors and like 10% errors typically. Okay, so like if this is the genome, so you can see that there are exons, introns, repetitive sequences, intergenic regions, right? So in case of repeats, if I am using like this short repeat, short read, okay, so you can see that this read is coming within the repeat. Okay, so if the repeat is say like 80 AG, 80 AG, 80 AG, you will not know where it is starting and where it is ending. So you can fit this read then this place or that place. Okay, you will be confused that where to, like how to assemble it, because all reads which are coming from this region look identical. Okay, but you have to cover the entire region. Okay, so you'll never know that how much was the length of that repeat region, and how to assemble that. Okay, so in this case long reads are better, because for example in case of long reads, at least by a few reads, okay, even longer than this, okay, because in case of nanopore you can go like up to 150 kb or something. Okay, so that will actually cover the entire repeat region. So you will know that okay, this middle part is the repeat region in this read, and basically this is intron and exon and like that. And that also you can confirm by this intron exon thing by the transcriptomic information, because you always use the like transcriptome as an empirical evidence when you are assembling the genome. Okay, so producing long read definitely has an advantage to get the contiguity, right? So basically the typical factors that are important here are the genome size. If you are going for a very large genome size, like for example aloe vera, which was 16 GB, okay, or wheat, which is like 14 GB, 14 billion base pair. Human genome is 3 billion base pair, right? Bird like peacock or like similar chicken, etc., they are like 1.2 billion base pairs, right? Plants, you see like a banyan tree or a peepal tree, okay, they look very big in size, but actually the genome size is only like 500 MB, okay, whereas aloe vera is like 32 times more than that, which is like 16 GB in size. Whereas aloe vera looks a small plant, whereas the banyan tree is a huge tree and also survives for so long time. So basically this is all about the genome complexity thing that we have talked earlier, that the amount of DNA or basically length of DNA or size of the genome does not tell exactly that how complex the genome would be. Okay, it is not a direct matrix of that. Okay, so genome size matters here. Then the ploidy level, that if you are dealing with a haploid genome, diploid genome, tetraploid, triploid, okay, so that will is important to know that what coverage you should go for. And if your genome is very repetitive, for example in case of plants, which is almost like 70% and more repeats, and also heterozygosity, right? So in that case definitely the longer reads will play a major role, because that will help you to achieve the contiguity which you cannot achieve by other means, right? So for example Illumina is one of the sequencing platforms, and Illumina reads 150 base pair, 250 base pair, up to 300 base pair, right? So these are like typically used for sequencing. And so one way to sequence is like if you have the sequencer at your place, like NovoSeq, which is like very high throughput, or NextSeq, okay, or MySeq, depending upon your requirement. If it is available in your lab or in the institute as a facility, you can use that and buy consumables and make your own library and run it there. Okay, or you have to outsource it to a sequencing company which can do this job at a very reasonable price. Okay, so right, so but you in any case you will be using some platform and like some company consumables or technology, right? So Illumina is one. So they have provided a calculator at their site which is called like sequencing coverage calculator, which can if you feed the genome size, it can tell you that how much coverage you need, what resources or basically consumables are needed for that, right, based on the platform that you have selected. So you can give it a try and see that how much for any for a bacteria you can calculate 5 million base pair. Okay, for a typical eukaryote you can calculate 1 to 2 billion base pairs, like that, right? So you have to tell the input genome size, okay, target region or size, and what coverage you want, right? And based on that coverage, it will tell you that how many cycles and how much also you have to select the instrument to do this calculation, then how much will be the total consumables that you need for that. Okay, and it will be, you can download this information and this will help you to plan. Okay, so now again the same question that how much you should sequence, and even if you have completed the sequencing and you have done the assembly, you have now like analyzing the data, then you will know that okay, basically I need more data to cover it. So you can do that at that point of time, or you can at least give one draft assembly, or at that point of time do the analysis and then again redo and make another paper later. Okay, and basically the objective of Genome is also like publications, and also you have to submit the data at the public databases, because you will also be taking data from other places and for your comparative analysis, and similarly they will also use your data for their analysis, and this will also lead to your increasing number of citations, right? So if you sequence too much or like very high, then basically that will increase the chance that there are no missing piece. Okay, so basically this way I have done a huge depth of sequencing and there will be no missing piece, right? Or basically the missing pieces will be very less, right? So basically that will increase the chances that you cover the entire genome. Of course, like the other, and also like making libraries using multiple libraries, is it okay? So you should not produce all the data in one go, but you should make multiple libraries and then sequence. Similarly, the different insert sizes should be used. Okay, that what distance your paired and read is. So the insert size could be like 350, 450 base pair to 2 kb to 10 kb to 50 kb. Okay, so that will also help in the assembly process, right? And there will be some genome that will need higher amount of sequencing, for example plants, and although they are bigger, okay, but they are highly repetitive, and that is the reason, right? And there will be some which, and they could be more complex also, right? And I have not covered it here, but basically the quality of DNA also matters, okay, because in case of this nanopore or PacBio sequencing, basically you should have high quality DNA and high quality and high molecular weight, so that you can have you can produce longer reads there. Okay, so for example in case of nanopore, as I mentioned, that it should be high quality DNA and also long read, long fragment. Okay, that means high molecular weight. Okay, so that basically like average read length you will get higher in that case. So generally you should get 10 to 20 kb average read length, and the highest read could be like several hundred kbs. Okay, and but you may still have like 1 or 2 or 3 kb reads, right? And basically choosing the appropriate consumables, you can also get rid of the shorter reads. Okay, there is a short read depleter also which is available in nanopore, right? So basically 30 to 50x coverage may be a good idea. Okay, even like with long read, if you are just using it for like if you have done very high level of sequencing by Illumina, then like 15 to 20x is also okay. Okay, but 50x will ensure that you will get a very good contiguity. Okay, right? So basically there are several papers that have reviewed these things, and I will also tell you some case studies, right? And for example nanopore for human genome, which is 3 billion base pair, it tells, nanopore tells that minimum 30x coverage is required, okay, with this average read length 25 to 35 kb, which is again to achieve like the objective. So in case of resequencing, the objective is to find the polymorphisms or mutations which could be harmful, or some random rearrangements and like that. Okay, because you are looking for a possibility of a like disease prediction or something, right? And then when you have done the assembly, basically this read N50, okay, that plays an important parameter which tells the sequencing, the result of sequence. You can say that what contiguity you have achieved, right. So higher N50 means a better value, right. For example, in case of aloe vera, the genome size was like almost 16 GB, okay, and if you are generating like 120 or 125 GB data, so you are generating almost like 15x coverage, okay, and which is good or reasonable for this big genome. But if you are dealing with a smaller genome such as a banyan tree, which is 500 million base pair, or a 500 MB, or a peacock genome which is 1 million 1 GB. So basically you should definitely go for like at least this case it is 15x, but you should go for at least 30 to 35x, 30 to 50x, right. So this long read data will definitely help you when you are going for the hybrid assembly, and basically using multiple methods of sequencing is like hybrid sequencing approach, and then assembling that data is hybrid assembly approach that we will deal later, okay. And the high quality DNA thing, if you as we discussed, so basically the presence of polysaccharides, polyphenols, and there are several things in say in insects and plants, okay, which interfere with the sequencing process and lead to poor quality DNA when you are looking at the quality values, right. And may may basically cause a dysbiosis in the your total estimate for like how many runs you want to plan and like that, because you might be expecting like 20-25 GB data per run, but it may lead to only like few GBs or like that per run, okay. So that is one of the issues that you have to handle, right. And but if you are doing outsourcing, then basically you are running with a contract that okay, I want this much data and this much I will pay for that, and that is also a good idea, and then it's company responsibility to give you that much data, okay. So long read is important, and quality of DNA is also equally important. Then the concept of link read such as the 10x genomics, okay, or TEL-seq, basically there also basically you should need at least 38 to 56x data. 38 is a minimum that the assembler takes, okay, and 56 is kind of good quality or good amount that you need to achieve a good sequencing. So link reads, because you are fragmenting the DNA, okay, labeling each and every fragment separately, like this part, okay, and this part, and then assembly fragments, and then combining that fragments again later in the assembly, okay. So this increase the contiguity. So 10x is plus Illumina sequencing is one of the ways to achieve it, okay. And the software that you will use for assembly is Supernova, right. But Supernova also needs an estimate of the coverage, okay, and how much data it can handle. So it cannot handle like larger genomes, okay. That means it cannot handle more than 2.14 billion reads, okay. So that means you can do like 1 or 1.5 GB DNA up to GB genome with the 10x technology, right. So basically it needs almost 56x raw coverage, okay. That means you should have, whatever is your genome size, you should have 56x more data, okay. So if the genome is 1 GB, so you should have at least 50 to 60 GB of the sequencing sequence reads, right, of 150 base pairs Illumina. But like you can still work with 38x, that is the minimal, that will give you the assembly. And basically number of reads also matter, because when you are estimating the size of genome by various programs which we will do later, so basically there also the number of reads that you have generated matters, okay. Then basically if you have a lower representation of reads, then like you are using an approach for short read assembly such as De Bruijn graph. So there you will not be able to generate sufficient number of nodes and edges where to run through the nodes and edges to basically to get longer contigs, okay. So so that means you should have a very high coverage of or sufficient coverage of short reads so that you can achieve good quality contigs, okay, because the algorithm is like that. So it will make a an array of k-mers, and it will try to connect those k-mers. So basically each k-mer becomes a vertex in the de Bruijn graph, and then it tries to run through the overlapping k-mers and do this iteratively and generate the contigs, right. So basically what I am saying is that you should have sufficient number of fragments or to generate sufficient number of k-mers which you can be used for a good quality or contiguous amount of contigs, right, contiguous contigs, right. And in case of long read, because the reads are already very long, okay, so overlap is sufficient, a decent overlap of 1 kb, 2 kb or 3 kb in case of like long reads is good enough to basically concatenate the reads and make the contiguous assembly, okay. So so basically overlap layout consensus method is the method here which is used to generate the long read assembly. And the combination of long read and short read is a better idea. So there are some tools which tries to merge the two together at the initial stage, or what you can do is for any genome you are generating long reads and short reads. So you should try to assemble the long reads separately and short reads separately, okay, and then merge the assemblies, right. Other idea is that you can merge both the things together at initial stage, okay, but that may lead to more amount of calculations also, okay, that and that may have some advantage, but generally like assembling the two separately, okay, generating the statistics from here and here separately, okay, and then combining the two where basically this also plays a role in correcting the errors in the long reads, right, and that is called like genome polishing, that we will see later, right. So that is also an idea in a hybrid assembly approach, okay. So basically connecting the two you can make a contiguous assembly like this, right. So short reads cannot cover like complex repetitive regions, so that is the disadvantage of short read, and it is not sufficient to cover the large genomes completely, right. So for larger genome definitely you need long reads, okay, but they suffer from errors, and their cost is also higher. So that is why a combination of the two and an appropriate coverage of the two is very much needed to achieve a good quality assembly, okay. So that is the advantage of hybrid assembly, that you are using the information that of the long read, that is covering like longer parts of the genome, like for example in case of nanopore it is covering 100 kb or 200 kb like that. So and you can imagine that how much assembly you have to do in case of short read to achieve 200 kb. So your problem of assembling short reads is solved, but then your 200 kb that you have may be having several errors, so you have to map the short reads on the long read, that is 200 kb, okay, and then correct the positions based on the evidence from the short reads, right, and that is how the assembly, the hybrid assembly approach using both the reads is very advantageous, right. So basically polishing for accuracy using the short reads on the long reads is very important, and similarly you can do the reverse also, okay. Then there are situations that people have reported genomes, but when you analyze that you find that okay, basically the assembly had several errors and they have not corrected for errors, okay. It is not a polished genome where so there are programs available to correct those things also. So for example like when you map this raw reads on the scaffold, that means the assembled fragments of the genome, right. So you will see that basically for each fragment the reads that are mapping have a different base pair as to what you have seen in the scaffold or the final outcome of the assembly, right. So what you should do is like for like this is your scaffold. So when you are mapping the read, okay, so basically for example it is a 20x coverage. So basically there will be almost like 18 to 22 depends, like not exactly 20 will get each and every position and each and every base pair. So for example there are 20 reads for example here. So in the scaffold, assembled scaffold, it was A. In this read also it was A, okay, it was also A, but it was T, 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 T here. So basically most of the reads that is coming from that region has T at that position, whereas in your assembly you have reported A, right. So that is a error, right. So you have to take evidence from the counts of all the 4 base pair at that position from multiple reads that you are using, and then correcting the errors in the scaffold. Okay, so this kind of error correction is also important. So there are programs like SeqBug and other programs which do this job, right. So typically a hybrid assembly workflow looks like this. So you should have the shorted assembly Illumina, PacBio or Nanopore, right, long reads. So 60x coverage for example from Illumina, 30x from PacBio, just an example, right. Error rate is only 0.1%, and in this case the error rate is like up to 15%, which is very high. That means out of 100, 15 base pairs will be incorrect. So that is why you need correct base pairs from the Illumina side, right. Then you do the short read assembly, and also similarly do the high long read assembly by Kanu and say Plantus or ABIS or anything for Illumina, right. And then basically using this long read you can achieve the hybrid assembly, right. So long and short done separately and then combined together to achieve the hybrid assembly by DB2OLC, okay. And then you will use the post assembly correction. That means after you have done the assembly you can use the short reads and long reads as the evidence, okay. So using polishing with long reads and polishing with short reads, so that you can correct the entire assembly for the contiguity and the base pairs that are there, okay. And then again you can merge the assemblies, right. And so basically either you can merge it at this level, right, or you can merge it at this level, right. And again you can do another round of polishing using both the reads, both the type of reads, right, to achieve even the assembly that you have generated is now rechecked for any errors and become better and better. So basically the objective is to get a high quality assembly with a free of errors, because you will be using this information to calculate the unique substitutions involved in evolution, or things like that, which will be used for telling that if the genome is evolved or not, and at that time these errors will become mutations or amino substitution that will lead to false results, okay. So genome assembly using long and short reads, and then basically this transcriptome. So parallelly with long and short reads you have also done the transcriptome assembly. The purpose was to detect the genes, their expression, but the same reads, short reads that you have generated from there can be used now here as empirical evidence to tell that the gene that you are predicting by Maker or any other software, right, and also during the assembly process. So because a transcript also contain reads from the genic regions, right. So that can be again mapped to the genic regions to fill the gaps, right, or basically to any genomic region. So this will also help you to improve this assembly quality. So transcriptome assembly by Agouti can be done, and then basically either the assembled transcriptome or the reads from that can be used for the hybrid genome assembly, okay. So some case studies I will present to just to give you a good idea. So like this is a genome of riverbank grape, this you may have seen, right. So all the basically the good or important genomes are mostly attempted and done so far. So you can see that they generated long reads by PacBio, which was 118x, which is very high. Then 10x link reads also they generated, 10x chromium, okay, 107x Illumina, like two different insert sizes, 406,000 mate pair sequence, right, 1640x, total 330x 1x genome they covered. Okay, of the genome size was only 500 MB. Okay, so you can imagine they have done almost 160 GB of data, right, and then their N50 that they achieve is 964 KB, which I think is lesser, because these days the like for this quality draft genome, the N50 should be about 2 MB and more, right. Number of pieces were 174, which is good number. Okay, that means they could the genome they could cover almost completely. Okay, Busco completeness 95%, that means almost 95% of the transcripts or genic information has been covered. That means that much genome has been covered, about 95-96%. And number of pieces, like breadth of genome, that was the question earlier, so it is only in 174 pieces. That means they have covered the kind of complete genome. Okay, then this is another like honeybee genome, 225 million base pairs, N50 is 3 KB, right, and longest was like 13 MB, and mean was basically this 5 MB. Okay, right, so N50 is 5 MB in this case, right. So 5 MB is a good quality genome, and for this they have generated like 40x PacBio data, okay, and 150x 10x genomics data. Okay, so long read plus short reads, hybrid assembly approach. So this is one of the fish genome. So it includes a paired end sequencing, mate pair of 4.5 KB and 7 KB, so different insert sizes, okay, just to increase the contiguity and more evidence of long distance connections, right, which helps in assembly, right. And link read data 10x genomics, about 78x, right. So in total they could get by this assembly approach using paired end, mate pair, 10x, and MNIN, it was less, basically 2.4x only, right, which should be more to get even better quality, but still they could get about 2 million base pair N50, right, which is a good number. Okay, and the assembled genome size in this case was 790 MB, all right. Then this is another medium sized tree which is available in China, Melania oleifera. Okay, and they also use the hybrid assembly approach using PacBio, long reads, and 10x genomics, right. And they also got a good N50. Sorry, this is the N50, right, of 4 MB, which is a good number, right. So whale genome, so they used Illumina data and link read data. Okay, so here they have not used the long read sequence, but they only use the mate pair, link reads and the link read, that is 10x genomics, and the short read data, and they could get a N50 of this number, which is 19 MB, right. So which is a good N50 in this case, and Busco completeness was 90.66 in case of complete genes, and including fragmented genes about 95%. And basically the higher the Busco value, more confident you are that you have covered most of the genes, and also it tells you about the breadth of the genome, that how much genome you have covered. It's an indicator of that, because if you have covered all the genes or most of the genes, that means you have covered all the chromosomal regions, right. Then in case of aloe vera genome, a combination of Illumina reads and the nanopore read, and like basically here earlier we are talking about 0.5 MB genome, it is 32 times more, like 16 GB genome, right. So basically 0.5 GB versus 16 GB, right. So it will be expensive project, and so basically total length N50 they got was only like 16 KB, but still it was good enough for a first draft, because it's a very large genome, and maybe among the largest genomes available on earth. turmeric genome, which is about 1.3 GB in size. Okay, so basically combination of link reads and nanopore reads were used, and to cover the genome of about 1 billion base pairs, and N50 value was about 100 KB, which is lesser because it is slightly older genome, but okay, so but and the BOOSCO completeness was 92%, right, in this case. In case of banyan tree which and Ficus religiosa and Bengalensis, people and banyan tree. So basically genome sizes were similar, like about 392 MB and 332 million base pairs, and N50 achieved was 486 and 553 MB, and basically the sequencing coverage for both were like 125x of 10x and 21x of nanopore, which is a good number, and that is why like the N50 was also reasonable, 0.5 MB, and also good coverage of the genome of the BOOSCO completeness. Okay, lastly, like this example of jamun tree, which is about 700 billion base pair genome size and a combination of 165x of 10x and 20x nanopore, which is kind of reasonable to get a good quality assembly. But here the assembly was about 180 KB, and we have in the last one we have seen about 500 KB assembly, which is better one, right? And but the BOOSCO completeness was 98.3, which tells that most of the genes of the genome are covered. Okay, then AMLA genome, which is 500 MB, combination of 237x 10x genomics link reads and 32x nanopore reads were used, and basically the N50 was 0.5 MB, not 597 KB, which is a good number, and BOOSCO completeness was also good, right? So you can see that in these examples I have told you that a combination of either long read, short read, or link read, short read, okay, are used to or basically long read, short read are used to cover the genome, or a combination of all three also, including the mate pair information. Okay, and this is an example of resequencing of Peacock. So basically you can see that over a period of time, in a few years, the assembly quality improved from, say, like there were 98,000 pieces, and now in the latest version it is only 69 pieces, right? So high quality genome with 444x Illumina, 81x link reads, and 7 to 8x nanopore, because in case of birds, producing the nanopore data is difficult because there are some technology issues and there are some contamination in the DNA that interferes with the sequencing process, and that is why you cannot achieve high coverage with the nanopore, right? So basically one flow cell can produce almost 20 GB, but in case of in this case, in case of birds, about 500 MB or like 1 GB is also an achievement for a bird's flow cell, right? So and you can see that higher quality assembly also provided good information about the number of genes from 15 KB, 15,000 to 26,000. Okay, the number of genes also increased, and correct estimate of repeats. So a better quality genome always will tell you will be in lesser number of pieces, better quality, and also will be more reliable in terms of gene number or repetitive content, and further comparative analysis, right? This is an example of resequencing of tiger, because tiger already was done by China, and then again it was resequenced at 27x, and it was sufficient to basically tell that the previous assembly had errors, and you can see that in the revised version 11,000 base pairs were corrected, that means of which 6,000 were in the transcripts or the coding regions, okay, and belonging to 4,000 or more genes, and so this was important things when you the errors in the transcriptome or the transcripts or the genes, which can be corrected by resequencing. Okay, so basically in this lecture we looked at the different ways in which we can estimate the coverage that is needed for any genome, and also for the different genome means, like it could be plant, which could be repeat rich, or bird, or okay, which has issues with long read sequencing, or basically the animal genome such as tiger or cow, etc., right? And of the different platforms, including the short read, long read, that how much combination of these is good enough to achieve a good quality assembly, right? And the importance of hybrid assembly approaches in achieving a good quality genome. Okay, so this is good for today. Okay, thank you.